So to begin my research, I used many materials. The first being a long Q-tip type swab. I used those Petri plates as you can see in the bottom right, but I filled my plates with non-specialized seaweed agar and my plates were also plastic. I kept all of my plates taped together in a biohazard bag after I swabbed the hoof each time. And as you can see in the top right, there is a photo of the sole of a horse hoof. And the V shape in the center is called the frog, and I swiped around the outer edge of that piece of the hoof. So here you can see that I began swabbing the hooves, and although this is a stock photo, this is very similar to the way that I was swabbing the hooves. First, I would pick them out with what you can see the person is holding is a green handle type thing with a brush on the top. At the point that is against the foot of the horse is a metal curved pick type thing, and you just pick out all the dirt or the feces or any packed material and then brush it out. After that, I would put the hoof pick down. I would kneel beside the horse and prop its foreleg on my knee. Then I would carefully take a swab and open the bottom portion of the packet so that I wouldn't contaminate the front cotton portion of the swab. I would then get my plate out and open it slightly to try and prevent any outside microbes from getting into the plate due to airflow. And then I would swab the frog and quickly plate the swab on the plate. So here you can see all the horses that I swabbed. We have Azrael, Kessler, and Robin. Azrael is my gelding, so he was the easiest one to start with. His sample has common similarities with all the other ones. Pretty much every sample had E. coli, Sarcina, and Enterobacter. However, only Kessler and Azzy had Bacillus. Azrael had a difference between all three. He had Cryptosporidium. Kessler also had a difference, which was Actinobacteria, but Robin, however, had the largest range of microbes, and his contained strep, microsporidium, and a gram-positive cocci that myself and Dr. Walker were unable to identify. So here's a quick comparative slide just because I know that not everyone is familiar with horse terminology, so I wanted to include some photos to help educate you. So shod means that a horse is wearing a shoe or has been shod. So the bottom photos on both the left and right show the frog in great detail, as well as the shoe placement on the hoof. And then the right photo on the bottom shows the shoes applied to the foot. And you can see as the horse is standing, you can see the hoof. And then the bottom, very little lip there is the shoe. So then the top photos are of a barefoot horse, which is how Azzy goes about his regular daily basis. And the left top photo, you can see the yellow line indicates the frog. And although it's a little dirty, that hoof does not have a shoe. So this was my favorite portion of my research project. And what was so cool is I did not have to incubate these plates for them to grow. And that is so cool because the microbes live in a natural environment within the hoof of the horse and the surrounding soil. So I was able to grow these in my room in their biohazard bag with no incubation whatsoever. And the microbes, as you can see, flourish on the agar. I have to admit that this portion of the project was also really cool, but it was extremely time consuming going through the staining process. because you have to apply several different chemicals for a certain amount of time, and then you have to either pour off or wash off, and you have to be very careful which step it is that you end up doing, because if you do one in the wrong order, then you have to completely restart your stain. And so after I stained, I had to fix the slide, and then I had to adjust the microscope several times under different lenses, and then add oil to be able to obtain the photos that you see in this PowerPoint. So here I included photos from Bing that I obtained through the PowerPoint program of each separate type of bacterium. Under Kessler's culture, the left is Actinobacteria. As you can see, it is gram-positive. On the right, there is Enterobacter, which is gram-negative. Azriel's culture example is Cryptosporidium. They are the flower petal-looking spore-type objects in the photo. Dr. Walker and I really were not sure what those were the first time we saw them and thought they were pretty interesting, so luckily we were able to identify them. In Robin's culture, he had the most. He had the strep, which is that first photo. Then he had Microsporidia, which is the black and white photo. And then he had the unknown gram-positive cocci, which could have been any number of things. I would like to mention that Kessler was kept in a pasture like Azriel, but he had sheaths on, so he was shod. From a medical standpoint, this is going to be the most interesting portion of this presentation because it deals with how these microbes affect these equines and their human counterparts. And for someone who's around these animals every single day, it was pretty shocking, especially considering that bacillus can cause anthrax, which is a very harmful disease to humans. And it can cause food poisoning, which I wasn't really surprised about because so many different microbes can cause food poisoning. Even E. coli can cause food poisoning. Now, E. coli was not a surprise to me at all whatsoever because of the fact that they walk around in their own feces. But I was surprised that it causes so many different inflictions in a horse, such as the neonatal septicemia of the foal, septic arthritis. I was really surprised by the mammary gland mastitis. Peritonitis was kind of interesting to find out. And then in humans, it causes hemolytic uremic syndrome, which is really crazy and affects the way that our blood cells function. And then that in turn obviously can cause kidney failure. From a medical standpoint, this is going to be the most interesting portion of this presentation because so this sample was a little disturbing considering the fact that this is the horse that I see almost every single day on a daily basis. He is my child and I take care of him on a regular basis. So in his sample, I found E. coli, which, as previously mentioned, causes a lot of different afflictions in horses, like the septic arthritis, peritonitis, and the septicemia. And then in humans, the hemolytic uremic syndrome, kidney failure. And as you can see, there's a photo of the peritonitis, which is inflammation of the peritoneal lining of the abdomen of humans. So that's what that is showing. Then there's the bacillus, which really doesn't affect horses. And that doesn't really surprise me either because of the fact that they really should be immune to some sorts of pathogens because of the fact that they do live outdoors and they do walk around in dirt and they roll in dirt and they eat off the ground. But in humans, it causes anthrax, as previously mentioned. It can also cause food poisoning, and then the septicemia and various infections, such as infections of cuts or lesions. It can infect your urinary tract, your ears, and your GI tract. I've included a photo of the bacillus. And then I have never heard nor seen Cryptosporidium. I included a photo of a slide of gastrointestinal cell, which shows various areas of infiltration, I suppose, and unhealthy pieces of the tissue. So cryptosporidium is a zoonotic disease, which means it can go in between animals and humans and vice versa. It causes diarrhea, dehydration, and gastrointestinal disease in people, which honestly kind of sounds like food poisoning, but the gastrointestinal disease can probably also lead to cancer. So this sample was a little disturbing considering the fact that this is the horse that out of every single one, and I honestly believe it is because he is barefoot. Because he's barefoot, the dirt and any caked in matter can easily escape the hoof and can be knocked out while he's walking or running around or being ridden. Now, Robin's culture kind of disturbed me a bit because I found strep in it, which I do not like. I've had my tonsils removed because of it, so I was really unhappy to find this, but also really happy for research purposes that I was able to find it. But strep can cause, in horses, it can cause infections in cuts or lesions. It can cause respiratory tract infections. And in humans, it disturbingly can cause meningitis and bacteremia. Meningitis is the swelling of the tissues of the brain. Bacteremia is a blood infection where bacteria gains access to the bloodstream. Next, I had never heard of microsporidium, but I found it in Robin's culture, and it often causes infection in horses through oral ingestion and gains access to the digestive tract. And then in humans, it causes encephalitis, which is swelling of the brain or the head technically, but it's pertaining to the brain, and you can see a photo of it down below the text description. And then in humans, it can also cause kidney infection, infections of the lungs, eyes, even muscle tissue, and the intestines. That portion about the intestines didn't surprise me because it causes the same infection in horses. And then I also found E. coli in Robin's sample, which didn't surprise me either because I found it in the two other samples. And like I said before, they walk around in their own feces, so I wasn't really surprised. And just to restate, it can cause food poisoning in humans, peritonitis, which is the inflammation of the peritoneal lining of the abdominal cavity. And in horses, it causes neonatal septicemia of the foal. It can cause arthritis and peritonitis, just the same as humans. Now, next, thank you so much for viewing my presentation. I hope that it was interesting, and I hope you learned a lot about the bacteria that might be in your horse's feet. Thank you guys, and have a wonderful week. Please stay safe, and God bless you.